focused around the question that's up in front of us on the screen, although obviously we're not the only ones trying to tackle this, and it's a pretty broad question. I'll get into one aspect that we're trying to address as I go through it, but I just want to say it's been an incredibly rewarding and fun experience to work with this group. This is a very interdisciplinary and collaborative group of researchers, and I think that is the exact right way of tackling something that is in some ways quite confusing. And I'll talk about potentially why what we're struggling to try to understand in food allergy. Okay, so let me start. And I'm going to start pretty broad just to set the stage in terms of trying to understand just allergy as a whole and what is confusing about allergy. And that takes us back to trying to understand just the immune system as a whole. And so I think our classic way of understanding the immune system, and especially in this day and age of COVID and other pathogens, is immunity to pathogens, right? So this is what protects us from the bad things out there in the world, like these respiratory viruses, gut bacteria, other types of infections. And so that's a classic view of what the immune system does, and it does it quite well. But when the immune system gets activated, it can also get activated to things we don't potentially want it to get activated to. Classic example of that, where I think we've really started to understand fundamental pathways that drive the immune system inappropriately, is in autoimmunity. So this is when the immune system turns against us and hits our own tissues. So example of this here is this person is attacking their own kidney. And so that's an example of autoimmunity. Type 1 diabetes, lupus, all of these are examples of autoimmune diseases. A more complicated type of immunity, which I won't spend any time on, but just to mention, is called alloimmunity. And so that's when the immune system still targets something we don't want to, but it's potentially appropriate in that it's targeting something that's been transplanted into our body. So in this case, maybe a donor kidney was given to this person, and it's being recognized as foreign and attacked just the same way the immune system might attack something like a pathogen. So in kind of contrast to all three of these, allergy in some ways is best defined by negatives. It's not an immune response to a pathogen. It's not an autoreactive response. It's not an alloreactive response. We're not reacting to things that are almost self, but just a little bit different. It's typically to things in our environment, substances in our environment. Here we have peanut, and here we have some kind of maybe environmental trigger, right? And this causes things like asthma, hay fever, food allergy. But it's a very unusual type of immune reaction that I would argue actually of all of these four types, we understand the least well. And so what that necessitates, and I think this is something that Fossey's really tackling, is understanding fundamental basic principles of the immune system to understand why and how this happens, which in many ways still is a mystery.
And so Stephanie, just thinking about that, when I look at that slide, which is very helpful to categorize it, it's almost as if the body thinks it's helping us by having a food allergy, right?
Absolutely.
Trying to protect us.
Okay. Yes. I mean, we don't try to personalize what the immune system is doing, but the immune system is set up with incredible numbers of checks and balances to make sure we don't have inappropriate immune responses. And that's, I would argue, why thankfully most of us don't have autoimmunity. We don't have allergy, right? There are many, many, many layers and steps, and I'll get into this in one of my slides in kind of more of a kitschy way, but why it takes a number of different types of hits to get an immune response to something we shouldn't. The immune system really is only going to get activated when it thinks it's when it's protecting us from something that is very analogous to trying to protect us from a pathogen. So I think you're absolutely right about that.
Thank you.
Another mystery in allergy is that what makes an allergen an allergen? I'm giving you a long list here on the right-hand side that is still only a partial list, but we know, for example, in food, there's the big 8, and now I think we're expanding out to the big 10. There's things like house dust mite, which is a important trigger in asthma. There's venoms. All of these are allergic type reactions, or what we would call a type 2 allergic reaction. But these things are pretty different from each other, right? There are a few unifying themes to some of the allergens that we think about as maybe one of the reasons that the immune system is targeting them, i.e., just like what Chris said, triggering the immune system to try to protect us from whatever this thing is, right? One is that some allergens have enzymatic activity. So the thought is they come in and they're damaging our tissue. And so that may be something that triggers the immune system to try to protect us from that damage. They also can get across the epithelium, meaning they can get into our bodies because they are either small, they are stable, meaning they don't get broken down by, for example, for food in our stomach by acids and those kinds of things. They're very soluble. I think this is an area that we're actually starting to explore. And if we have time at the end, we'll come back to. I think it's a very important piece of what makes an allergen. But I would say still to this day, this is a mystery. And so in thinking now more specifically about what happens to food, I want to very specifically focus our attention on food allergy. Because there's a lot of different types of adverse reactions to food. And what we're really trying to uncover is the basic mechanisms of the allergic or atopic response to food. And so that's to things like I've already pointed out the big 8, where you have a reaction, whether it's associated with other factors going on in the environment or not. And there's other parts of allergic reactions that are slightly different from what we think of as the classic food allergy, but still are allergic responses to food. A good example of that is eosinophilic esophagitis, or EOE. In contrast, there's a number of other types of adverse reactions to food that are also mysteries, but some more than others, where we're having a bad reaction to food, but it is not a food allergy. I think a classic example of this is milk intolerance. So this is an example where as we age, evolutionarily, we've lost the enzymes that help us break down milk, in particular, lactose, the sugar in milk. And so that causes problems in our guts, but that's not an allergic reaction, even if it has some, negative manifestations in our guts. Gluten intolerance is another good one. And in contrast to that, celiac disease, which is not gluten intolerance, but actually a form of a autoimmune disease where you target your own molecules, but only in the presence of gluten, is another form of an adverse reaction to food, but is not exactly a food allergy. So really now I'm kind of laser focusing us in to understanding this top piece. That's what our work, and I think what Fosse's rightly focusing on to understand. So how do we do this? I think in general for the lay public and for scientists, it's a slow process, slower than we would like. But I would argue the best way of understanding best how to develop new technologies to treat or to diagnose or to intervene in food allergy is to really mechanistically understand what's going on, identify fundamental principles of what the immune system is doing, and then apply that. And actually, I think people outside of science don't realize how creative science can be, especially in group settings, like where you have scientists who are all thinking about a problem from different vantage points. And so, you have to be creative to start undiscovering things that you might not a priori already be thinking about. And that's the way we do this in my lab is we try to understand this by going back and forth between human studies. So this is getting samples or clinical data from patients with food allergy and going back and forth between a mouse model or an animal model to try to test particular pathways and really get mechanistic understanding. And just from this very short list at the bottom, you can see that there's already a number of different things that have been proposed as factors that are mechanistically important for food allergy. We are thinking about just 2 of these, DOCK8 deficiency and IgA deficiency, and I will really focus in today on talking about IgA. But you can see how many of them are here. And that, I think, highlights why this takes some time, but also it's a very complex puzzle. This is not like a 10-piece puzzle where you figure out the 10 pieces and you have a pretty simple picture. It's more like a 1,000-piece puzzle, and we have to really figure them out as we're going along. And so my kitschy kind of example for this is on the right-hand side, I love cake. So on the right-hand side, this is a very easy, quick way of making a cake. You essentially just add water, maybe 1 or 2 other ingredients, and you get a cake. And to get allergy, I would argue it's really not just add water. There's a number of different ingredients in the right time at the right place that have to come together for you to get a cake in this example. And so again, the way of thinking about this for allergy, the recipe, if you will, is that it's environment, it's genetic factors, and it's a bunch of question marks that all of us are trying to identify. And so that comes together in a cake that looks like this, right? In an unfortunate disease state where you get, the timing, the kinetics, the dose of each of these put together in a particular order come together to unfortunately drive this disease. And so all of us are really tackling this as a group from different vantage points, different angles. Some people are thinking about the genetics. Some people are trying to identify microbiome aspects that drive this. We are thinking a lot about allergen exposure. So over here on the egg side, trying to understand how this works. And so that's what I'll focus on just for the next couple of minutes in terms of the fundamental questions we're trying to address. And so these are the two big questions that drive multiple projects in the lab. I'm only going to speak about one of them today. And that's, why do some children develop IgE to allergens? We would call this sensitized. Why are they sensitized to a food allergen? So I will not get into too much nomenclature and complex jargon that immunologists love. I realize it's sometimes quite problematic. But the take-home point here is there's antibodies, these proteins that are typically in our blood, but also in other sites in the body, that drive different types of immune reactions. IgE is the classic antibody that we think about with allergy. And so when you have this antibody, you are what we call sensitized. And IgE is a very rare antibody. Actually, most people have almost none, and the people with allergy, actually really strong allergy even, still have very low levels. It's a very, very rare antibody, but it's quite powerful. And I think the converse of understanding sensitization is, how can we promote tolerance? And tolerance really means the immune system fails to react to an allergen, right? We think about tolerance in many different disease states where we're trying to undo the immune system, really, like autoimmunity. We want people to be tolerant to their own body. And in this case, for allergy, we want them to be tolerant to allergens. And what we don't want to have happen is this. So these IgE antibodies get put on a cell type called a mast cell that's all over your body, in particular in mucosal surfaces, so that when this peanut allergen comes in, it activates the cell. And when the cell gets activated, it induces a pretty strong reaction up to the point of what we call anaphylaxis, which can be life-threatening. And that is manifested essentially across your entire body, from your skin to your airways to your guts. So this is a pretty, like I said, very potent aspect of the immune system. So the clue that tripped off this kind of project that I'm going to get now more detailed into was thinking about actually a combination of those two pieces that I just defined. And the reason that we know about this is that for any of you who have an allergy or have been worked up for an allergy, there's really two ways we try to diagnose allergy, in addition to a really good clinical history. We either do something called a skin prick test, which is pictured here, where you inject the allergen under the skin and you look for what I just kind of showed you in a diagrammatic form. You look for a reaction. The other way is you give some blood and they look for IgE antibodies in that blood. And this is an example of a machine, for example, in a clinical immunology lab, which is where my clinical background is. And either of those tell you that you've made an IgE response. So that's the sensitization. So this is actually an incredibly sensitive method for detecting whether or not you've made IgE. And what it can tell you is that you've had this reaction. You're sensitized. What it cannot tell you, though, is whether or not you're likely to even have a reaction at all, or if you have a reaction, how strong it's going to be. It gives you a probability. And that's why, unfortunately, in food allergy, we're really stuck with oral food challenges, where you get one of these results back or both, and they're positive. And then, to be really sure, you have to undergo eating that allergen. So that can be a very frightening process. But the clinical data that really intrigued us, that we thought would be really helpful to dive into and understand better, is that there's an interesting phenomenon in some people called sensitized tolerance. And that means you have an IgE antibody, but you can tolerate the food just fine. You don't have any immune reaction. And so that led us to start thinking about, well, what are some other ways the immune system might be reacting to the allergens that would actually block this harmful effect of IgE, this mast cell degranulation arm of the allergic response? So that makes us start thinking about different types of antibodies. Antibodies come in many flavors. I will just talk about one other flavor, so you don't have to, we're not going to go into all the mechanisms of how these different isotypes happen, which is what we call them. But I want to really focus on one other isotype of antibody called IgA. And so the theory that has been out there actually for quite some time is that IgA is an antibody at your mucosal surfaces. So it's in your lungs, it's in your nose, it's in your gut. It may actually be able to induce tolerance. And that's what we set out to really study. And so the idea would be something like this in general. And now I'm going to focus in on IgA. In sensitized tolerance, you have the food allergen around, you have IgE, but they don't see each other. And that's a really fundamental piece that I think we we don't understand very well. And that's something my lab is very interested in understanding. With someone who has symptomatic food allergy, the allergen here, peanut, gets across and triggers this reaction that I've been talking about. And so the idea,
and Stephanie, just so our audience knows that this is what this cellular gut lining. So this is when you eat food on the inside, and this is the outside. So, and this is the gut lining.
Thank you, Chris, for reminding me. I should have explained this better. This is something you've eaten, and so it's inside of the gut lumen, inside your gut. And this is the single cell barrier between what you've eaten and the rest of your body, including your immune system. And this is that mast cell sitting there in it's called the lamina propria, but it's really the inside of your body, inside the guts. And then this is trying to diagram a blood vessel, so that actually the allergen here, peanut, is not just getting to this mast cell, but actually might become systemic, meaning you get it across your entire body. And so the idea here is that IgA in the gut, actually in the lumen here, in this white space, has been shown to really block a lot of different things that we don't want inside our body. For example, pathogens, toxins, and actually certain types of bacteria. And the question was, does it actually regulate food allergens too? And so the reason this is important is, if that's true, IgA could be a really powerful biomarker. It could be something we could use to diagnose somebody who is sensitized tolerant, meaning they could eat peanut even though they might have this blood test or this skin prick test that has IgE in it. And the other part of it is, if it's also, if this is true, the idea here would be, could we induce IgA so that it's doing this in patients who are already on the right-hand side here symptomatic? And so this is why we really set to understand this at a deep level. And we spent a couple of years trying to answer this question, and I'm going to summarize it. I'm going to skip, just in the interest of time, that other people besides us have been thinking about this for a while. I'm going to summarize a very long piece of work from an incredibly talented allergist, Elise Liu, to summarize what our findings were. And they were really surprising. Despite what people had been thinking about in the field, we found that people with and without peanut allergy had essentially the same levels of IgA to peanut. And that's both in the blood and in the gut. And that was not what we expected to find. And this is how science goes. This has ruled out, I think, one hypothesis that we had, which was that IgA was here in the lumen, preventing this allergen from getting across, and could explain sensitized tolerant. And the reason that's important is now we're moving on to the next phase of this, to understand, okay, is there something, if it's not levels, if it's not quantity, is it something special about the IgA that someone makes who's sensitized tolerant versus something that's missing in somebody who has food allergy? And there's some reasons for us to think about this, but maybe I'll wait until the Q&A session for us to talk a little bit more about that. All right, I will just end by thanking my incredible team. This is my own lab team, which has actually spanned both Yale and Northwestern, and has been really trying to answer some of these fundamental questions for almost a decade now. And in particular, I already highlighted Elise Liu's work, but there's really an incredible group that works on food allergy, tackling this question and from many different vantage points. And again, with the help of FASI to do this, and the incredible group there. So maybe I'll stop now and answer some questions, including maybe what other thoughts we have about IgA in this.
That was a great talk. So thank you so much. And there are so many questions that I'm thinking of, but if people have some questions, please feel free to put them in the Q&A, and we can ask Dr. Eisenbarth. And, the first question is, because it's really interesting, to think about IgA, and could we potentially use this to stop the reaction between the peanuts and the gut lining. And you had mentioned some, looking at different types of IgA. How do you think this could be used, to help us as a biomarker for food allergies? Is there a way to think about it as a biomarker to help us get maybe more specific diagnostics, or for therapeutics?
Yeah. So that is indeed where we started. The easiest would have been if someone with food allergy who had symptoms had no peanut specific IgA. We were measuring very specific IgA to either peanut or egg. That was not true. So that would have been a very easy yes, no, like this person has IgA. We should, be more inclined to let them trial the food that they potentially are sensitized to see if they are actually tolerant, right? Because that simple assay that we've looked at so far is not giving us that hint. We're starting to think about other aspects of IgA that may be important for protecting and actually in part with direct help from FOSSI, trying to find collaborators who can help us try to identify, is the form of the IgA different that might be protective? Meaning there's sugars that can get added to antibodies, and there's other ways that antibodies can be functionally different between people, even if the levels are the same. And so we're trying to get at that, because that would be potentially a pretty tractable assay that could be used clinically, in addition to the other kinds of tests that are now being used to say, what's your risk of anaphylaxis? Are you more likely to react, or are you less likely to react? And I think that would be incredibly important and helpful. And that's where exactly we're going. And some of the hints already that may be relevant is when people undergo something called oral immunotherapy to treat food allergy, IgA levels have been shown to go up. So maybe there's some kind of IgA that can be protective. And our goal now is to find what that is.
Yeah, well, I mean, I guess the question is it protective or is it a defense mechanism that's going up as well? And so that would be probably hard to know, and be interesting to think, does it change over time? Like, is your IgA different when you're a newborn, when you're more susceptible to sensitization versus when you get older, and how you can look at that? So many good questions. We have another question in the chat, and I'm going to be very cognizant of the time because I know we only have 30 minutes here. In addition to IgA, what other factors do you think could be inducing this sensitized tolerance? Big question.
Yes, great question. Can you
Yeah.
still see my slides?
Yes, we can still see it. Yeah.
I did focus in because I wanted to give you one example of some research we've been doing in the last couple of years, very concretely. But this is a broader question than just IgA. So this is really the framework we're using to try to understand this exact question that Carlos brought up about sensitized tolerance. I believe that there are pathways that are regulating how these allergens get across. IgA may be part of it, but I think there's going to be other aspects to this that regulate the access, really, of the allergen to the inside of our body, to the immune system in our body. And so that's actually where we're focusing in parallel now on characterizing other aspects of IgA, while also characterizing other aspects of the immune system and the gut itself, to say what might be different about how the gut handles allergens, and either does here on the right-hand side or does not let them across. I think that's a fundamental question. We don't really very well understand, actually, for allergens or even food proteins in general. So that's exactly where we're going.
Our annual symposium this year will be on immune tolerance, and that'll be really exciting to bring everyone together to talk about that and really hone in on finding that. And a lot of the basic foundational work really had to be done to even get this far, to be able to have these direct conversations, which is very important, which is probably a lot of what FOSSI initially really funded. So now we can really get down into it. I love this question. Someone said, how can parents with children with allergies individuals help? Clinical trial participation, donations, etc. Yes and yes. So participating in clinical trials is a really good thing. We have a trial out there right now that we're working with Mass General and Boston Children's, and hopefully CHOP will be on board soon too, looking at single cell analysis in the gut. And that really allows us to look at what the gut looks like in a child with food allergies compared to controls without. And so we'd be able to look at these questions of the IgA versus, is there different types of cells involved? And so really interesting thing. And donations, FOSSI is 100% philanthropically funded, and 100% of donations go directly into food allergy research. So that is always important as well. And so just one other quick question that we have about 1 minute. What other areas, I'd love to hear more about other areas of research that you're doing in your lab as well in the food allergy space. And it's super exciting, to hear about you starting this new center as well. And that's really great for the field of food allergies and the potential for those clinical trials to come out.
Yeah, and this relates to actually both of the last two questions. I have an incredible amount of respect for parents who enroll their children in trials to do these, because we really, I think mouse models are powerful. We can learn a lot about particular aspects of the immune system. But it's really important for us to go back. It's fundamental for us to go back and understand what is happening in the human immune system and in the human gut. And that is actually, unfortunately, an area that's very hard to access. So, the skin, we often get great samples from. Other aspects of the body, blood, we often have, we're readily able to do assays on and interrogate what happens in the immune system in those sites, right? The gut is a little bit privileged because we don't often get to see what's going on in the human side of that. And that's incredibly important. So that is something that we are aiming to do as well at Northwestern, and in collaboration with Fosse, do it very similar to what you guys are trying to do, to really try to get a deep look now at what's happening inside this kind of privileged site, if you will. As part of that, I focus on IgA because I'm focusing on the blocking side, potentially the inhibitory side, but we also need to understand what's tipping this whole process to happen in the first place. And so we, in my lab, think about both sides of that coin. So we're also trying to understand what triggers somebody, induces them to make this IgE in the first place. And we've discovered cells and pathways that we think are relevant for that. And if we understand that, again, we work our way backwards and say, okay, where's that first tipping point that we might be able to block so that we can change somebody's direction, their trajectory, after potentially manifesting early symptoms of allergy.
Yeah, and that's great. And those are all important questions. And when you were talking about the correlation between mechanistic between humans and mouse studies, when you think, like I'm a cancer doctor, that's how we had so many treatments for cancer, is having the correlation. And they're both super important to do. And from all the patients that were, gave to us by joining these trials and giving their samples and doing all that. And that's what makes a difference. And we'll get there.
Cancer is a great example of where we have access to the tissue that's relevant and we can study it, right? Not us personally, but the field. And that has transformed many different types of therapies, right?
Yeah. Someone asked about EOE. I just sent you a message that you can email us and we can tell you about some of our EOE, and we'll try to make sure we have one of our webinars focused on that. But due to our time, I just want to thank you, Stephanie, for joining us today. We're very grateful to have you and all your amazing research. And, you're quite a respected scientist in the community, and we appreciate you being part of the Fosse team. So thank you all for joining us. We really appreciate it and glad you're here today. And we really want to thank you all for supporting us and all that you've done for us in the Fosse community, because it's all of you that are helping us get to all these diagnostics and treatments for our friends and relatives. Have a good day.